thank you so much for the introduction, Kate, and for the opportunity to speak. I'm from a clinical microbiology and infectious diseases background, but today I'm going to talk to you about economics. So do bear with me, and apologies in advance to any economists if there are any amongst you. But this is, I'm currently doing a PhD which focuses more on ESBL colonization in human adults. But for this particular piece of work, I was interested in looking at economic evaluations of whole genome sequencing for surveillance, and particularly for surveillance of bacterial pathogens, and particularly thinking about how that would apply to resistance. So this is my sort of attempt to make my own understanding of economic evaluations. It's an umbrella term, economic evaluation, where on the one hand are considered costs, and on the other consequences or indeed benefits, they might also be called. And the costs could be of varying different types, and a simple costing analysis that considers no benefits would just be a costing analysis. It's just totting up what something's going to cost. But a more complete economic evaluation should take into account both sides of this equation. And the benefits that might be considered could be monetized or non-monetized, and depending on whether they are considered in a monetized form, such as a cost-benefit analysis, or a non-monetized form, which could be a cost-effectiveness or a cost-utility analysis. These are different evaluation structures. And I think it's important to say this up front, because I think this sort of speaks to why it's difficult to try to amalgamate this data in the form of a systematic review, because there are many ways to do this properly, and that can make it hard to compare across studies and settings, as I'll go on to explain. So what I set out to do here was a systematic review, where the inclusion criteria were that it should be a full or partial economic evaluation of whole genome sequencing being used as a surveillance tool. So not for diagnostics, for example, and not for a research purpose, but trying to either evaluate an actual or a possible surveillance situation. And I was interested in bacterial pathogens, excluding TB, focusing primarily on those which were in the WHO priority pathogens for AMR list. And the reasons for that was that I was interested in how this would apply to AMR, even if the particular pathogen wasn't resistant, by which I mean considering, for example, Enterobacterales, for example, but regardless of whether they were resistant or not, in terms of how that would work, what would the sequencing costs look like? Partially, this was regardless of their AMR status, because the studies that were specifically looking at AMR were very, very few. The time frame was a sort of slightly arbitrarily defined 1991, before sequencing was available, up until 2021, which is when we did the search and wrote the review. So I won't dwell on this too much, but to say search strategy yielded 681 hits, of which eventually whittled down to 9 included studies, which were selected for a narrative synthesis. And the quality of these was assessed using the Drummond-Jefferson checklist, which I found quite interesting, as a kind of, I think it was originally developed as a checklist to improve the quality of and reporting of economic evaluations in healthcare. So our findings. So the flags here represent how many times a country was represented in the included studies, and some studies included data from more than one country, in which case I have represented them all. So the number of times a flag appears is the number of times they appeared in a study. So four studies talked about Australian data, 3 the US, 2 each from the United Kingdom and Canada, and one study used some data from Italy and 1 from Argentina. So I think it's worth pointing out there, of course, that Argentina is the only country represented that is technically low and middle income, in the sense that Argentina is an upper middle income country. The rest are all high income settings. The contexts in which whole genome sequencing was being used as a surveillance tool for bacterial pathogens were either sort of food pathogen surveillance, so surveillance of diarrheal disease, but not necessarily hospitalization, really was what most of those were doing, or for far more hospital-focused infection prevention and control, so as a tool to aid infection prevention and control. So 4 were looking more from a sort of food safety surveillance, foodborne pathogen surveillance system, and 5 looking at hospital infection prevention and control, most of which were from the perspective of a hospital itself. 1 was from the perspective of a local government, but considering a hospital as the site. The cost per isolate was listed in all the studies and ranged from 72 to 194 2020 US dollars, and that was for the whole genome sequencing cost. Pathogens covered, so they had to have at least one of the WHO and priority pathogens, but they could have lots of different pathogens as well. So if others were included, if it was easy to totally exclude, for example, one study had a viral program, but it was a very extensively written up thing, so you could actually just chunk out the viral bit, but otherwise there were an array of different pathogens that were described as part of the surveillance systems. Some of the studies included these break-even analyses. So they didn't all include this, but I thought it was interesting to just quote what a few of these are, because I think it gives a flavour of how difficult it can be to compare results. So, for example, even the 2 studies that quoted a break-even point for salmonellosis, one of them was saying between 0.2 to 1.1% of cases of salmonellosis or a single death would need to be averted in order for that program to break even. Another study, it was 1.9%, but of all notified salmonella cases. Another study predicted to be cost effective, I think this was a hospital surveillance, as long as effectiveness greater than 30%. So these are sort of quite heterogeneous in the reporting. And I think it's also worth saying that this is also highly pathogen specific. So one of the studies really makes the point that a single case of listeriosis averted was enough to really justify the program, whereas for salmonella they would require a lot more averted cases. And so obviously that can make comparison across studies extremely challenging, and for that reason, meta-analysis not possible. So we went for the most simple way of trying to aggregate and make sense of these results, which was a vote count on the direction of effect. And all studies were broadly very supportive of the idea that whole genome sequencing was an economically viable method of surveillance, or an economically beneficial benefit of surveillance for the pathogens that were looked at. So the key takeaways from this piece of work were really that I think the available evidence does support the use of whole genome sequencing for pathogen surveillance on economic grounds for bacterial pathogens, but there wasn't a huge amount of evidence, and it was very biased to the global north, and much of it relies on modelled estimates. And I think at this point it's also worth saying, when we first conceived of this, that the main interest was in considering the role in AMR. And really, none of these studies were looking specifically from a standpoint of only looking at AMR pathogens. And so that's why we flexed a little bit to think, well, that similar sorts of pathogens, similar species, etc., how would the economics of that look? But looking specifically at AMR in and of itself, none of them did. And I think that would be perhaps a slightly different equation in terms of how would you measure some of those costs, how would you look at some of those benefits. So I think that's an interesting point. And I mean, this review and the search was in 2021. I haven't officially rerun all the searches, but a brief search last week suggests there hasn't been loads more that would have been included when I had a brief go at rerunning some of the search. I think another takeaway was if something's worth doing and worth doing right, comprehensive analysis different outcomes and more different inputs, and that made it a lot easier to see how that translated or didn't translate across studies. So useful to think about how studies are reported and how their quality can be maximized. Data from low and middle income settings are very much lacking here. And another point that I thought was quite interesting that was brought out in some of the conclusions of the papers was that modelling based on traditionally detected outbreaks will underestimate the benefits of whole genome sequencing because of whole genome sequencing's ability to detect outbreaks that would otherwise have gone undetected. So, for example, some of the hospital data that fed into some of these analyses was looking at outbreaks that they had detected by a variety of non-sequencing based means, and using the data from those outbreaks to model what would have been the cost savings, or the costs and cost savings had a whole genome sequencing surveillance been available. And making the point that if that's the only data that feeds the model, you'll underestimate the ability to have an undetected, an otherwise undetected outbreak detected and dealt with. So an interesting point there. So I will leave it there. Thank you so much. This was the paper we published. Should anyone want further information, also very happy to take questions at the end. And thank you to all the co-authors who contributed to this work and the institutions that supported it as well. And thank you to you all for listening.